Hi, and welcome to Everyday Potato, where everyone's a commentator. I'm your host, Jack Wiegand, and this is my guest, Jared King. Jared is a student of neuroscience at the University of Oregon, currently conducting research for the McNair Scholars Program. Would you like to tell us a little bit about your research?
Sure. First off, thanks for having me. So I am a fourth year student at the University of Oregon. I currently work in a lab at the Institute of Neuroscience there. And our lab is primarily concerned with olfaction, particularly in mice. Olfaction is your sense of smell. And so mice are known to be animals that use their sense of smell as their primary sense for locating things such as like food, finding a mate, various different things. And so we're interested in looking into some of these things. So some of the things that my lab does is, we have one person who is interested in how mice use their sense of smell to navigate. We have a rig that uses a camera to look at head angle and body angle. At the same time, these mice have been implanted with a thermistor in their nostrils. So we have a wire hooked to the thermistor, and as they're breathing in and out, the thermistor bead expands and contracts. We can see that on an oscilloscope as like an electrical signal, right? And so we watch their sniffing patterns along with their head rotation to see how they're going about locating, say, an odorant for a reward, maybe how they go to find food or whatever. Another thing we're looking at is how mice respond to various stimuli, how they respond, what are their sniffing patterns in response to odors that they find pleasant and unpleasant. That's my project. So I work on trying to establish different sniffing behaviors in response to positive and negative stimuli. The reason I'm interested in that is because we've done this research with humans, and we found that humans that come into contact with a positive smell typically increase their magnitude and their frequency of sniffing, which makes sense. You smell chocolate, you like the smell of chocolate, you're going to smell it more.
Coffee.
Right. And you can definitely see this with children, which is, for one paper led by a man by the name of Sobel, they performed this experiment on a bunch of elementary school-age children who could tell you if they liked to smell or if they disliked to smell. And as they were responding to these smells, like I was saying, they would get a positive smell, and they would increase frequency and magnitude. If they had a smell that was aversive, they would do the opposite. They would decrease their frequency and magnitude. So you're sort of trying to clean your palate out and hold your breath till you can get away from the source. Right. What was interesting is that they also performed the same experiment on children diagnosed with autism spectrum disorder. And so what they found when they did that was that in response to a positive stimulus, the children with autism spectrum disorder would respond the same way as the children they call typically developing. They would increase their frequency and magnitude. They're trying to get as much in as they can. But when they inhaled a stimulus that they thought was negative, they didn't change their sniffing behavior. They would either treat it as if it was clean air, or they would treat it as if it was a positive stimulus. So there's something going on. This is a very consistent finding, too. Now, how that relates to mice, we're not sure. A lot is known about autism as far as symptomology goes, but not a lot is known. We know some about causes. We don't know much about the mechanisms from those causes. So we know there's like genetic, environmental, different factors that can come in with autism spectrum disorder and create many different symptoms. And we treat symptoms, but we don't treat disorders. So in order to start understanding this disorder, we're kind of looking anywhere we can for these little mechanisms, right? And so we think we might have found something. And the way we'll know is we'll test with these mice that I'm working on. So as I'm studying what I call the typically developing mouse, I'm studying their response to positive and aversive odorants and seeing if they change their sniffing cycles. And if they do, then that kind of segues me into working with what I call mouse models for autism. Mouse models for autism are, that gets a little complicated. They're the FMR1 knockout mice.
Okay.
And what this means is that they have genes associated with Fragile X mental retardation syndrome.
Okay.
What that syndrome actually is a replication of a codon, I believe it's CGG, on the X chromosome in humans.
Okay.
And so when you replicate this codon more than 200 times, if you do it more than 50 times, you start having issues. You start having symptoms. Once you do it more than 200 times, you're genetically classified as Fragile X mental retardation syndrome, right? So the mice we're working with, they're not exactly the same, but they exhibit the same behaviors because with having this codon repeated so many times in this DNA sequence, it actually methylates the entire sequence, and so the sequence stops working.
Huh.
Right. So it no longer produces the FMR protein, which is what leads to
Right.
this disorder, the symptoms of this disorder. So in the FMR1 knockout mice, we can't exactly repeat this codon over and over again. Instead, we take it out completely. But it has the same overall effect. The FMR protein can't be replicated.
Huh.
So we get similar behaviors. And in fact, if you actually watch the mice, you can characterize their behaviors very similar to that of humans. So the reason we're using the FMR mice is because in humans, humans that have Fragile X mental retardation syndrome, about 46% of males are also diagnosed with autism spectrum disorder, and about 15% of females are diagnosed with autism spectrum disorder. And so it makes it really easy to use these mice because they're one of the only known single gene causes for autism. Exactly. So that's kind of the gist of everything I'm doing.
Okay.